All right, are there any questions? So then we move on to, I have a lot of material to cover today, all right? So we are going to move on to chapter 10 first, okay? That's about stomatal biology. So chapter 10. Right? Now we know that leaves have pores in them that are called stomata, right? So pores in leaves. So these are called stomata. And we know that carbon dioxide enters through the stomata, right? And water vapor goes out, right? So entry point for carbon dioxide and exit point for water. Right? Now this needs to be controlled, right? Like when the plant is going to do, are you having problems seeing anything or no? Which entry point, ENTRY. Yeah, just ask me, instead of straining, just ask me, I'll repeat it, yeah. That's the entry point for carbon dioxide and the exit point for water, okay? Now, that has to be regulated. Say, for example, if it's very dry, water shouldn't be lost, right? So then the stomatal pore should close, right? That's what the plants typically do, right? So how is that achieved, right? Now, that is achieved through what are called guard cells, right? That line the stomata, right? So each stomatal pore has two guard cells, right? So they look like this, right? There's one guard cell here, another one here, right? And in response to an external stimulus, right, the turgor pressure inside the guard cell will change, right? And that will cause the stomata either to open or close. If the pressure goes up, the stomata will open, right? So these are guard cells. So these are two guard cells, right? Two guard cells. So external stimuli that bring about changes in the guard cell turgor pressure cause the stomata to open and close. To open or close. Now, under normal conditions, light is the most dominant stimulus, right? That causes the stomata to open or close, right? Because if the plant is going to do photosynthesis, right, it needs to absorb carbon dioxide. Stomata need to open up, right? So light is the most dominant stimulus, right? So under normal circumstances, light is the most dominant stimulus that causes stomatal opening. And there are two components to this stimulus, right? So light brings about photosynthesis, right? So if there is photosynthesis in the guard cells, right, the cells that I drew over there, the guard cells of the stomata, right, that will cause stomatal opening, right? So one component is photosynthesis in the guard cell chloroplast. And the other component, right, that causes stomatal opening is response to blue light. So now if you want to see, blue light can also cause photosynthesis, all right? So if you want to study these two things in isolation, right, you can do it in two ways, okay? One way would be to put in something that is going to block photosynthesis. So you'll see only the effect of blue light, right? So these two responses can be separately studied by two different approaches. Right? The first one would be to block photosynthesis. So you can use a compound that is called dichloro phenyl dimethyl urea. I'm not asking you to memorize any of these names. Right? This is just for your information. But it's basically used to block photosynthesis. So DCMU. Right? That's the abbreviation for this. Right? So you can use this to block photosynthesis. Or more specifically, photosynthetic electron transport. Remember, we talked about the photosystem 2, photosystem 1 electron. This compound is going to block that electron transport. So photosynthetic electron transport, or the other thing that you can do is you want to study the effect of blue light. Okay, you can actually saturate the photosynthetic machinery using a different colored light. Let's say red light, right? And then you see if anything, if the stomata opens more when you bring in blue light into the picture, right? So the other approach will be saturate photosynthetic opening of stomata using red light, and then see how much of additional opening occurs in response to blue light. Now, how does blue light bring about stomatal opening, right? Blue light actually activates, so blue light activates a proton pump at the guard cell plasma membrane. So the guard cell is just like any other cell, right? It has a plasma membrane, right? So if you in the presence of blue light, hydrogen ions are pumped out, protons are pumped out. So blue light activates a proton pump at the guard cell plasma membrane. So when that happens, when protons are pumped out of the cell, right? What happens to the pH outside the cell? Does it go up or does it go down? It goes down, right? Higher hydrogen ion concentration. So this is going to lower, so lowers pH of the apoplastic space, right? That means the space that is made up of all the cell walls, okay, and the gaps in them surrounding the guard cells, right? So when that happens, right, potassium ions can enter the guard cells, right? Because outside it's positive or hydrogen ions, potassium is a positively charged ion, it can go down, right? So potassium ions can enter, right? Potassium ions enter a guard cell. So K plus enters guard cell through what are called potassium channels. Again, remember, potassium channels means those channels are things through which things can flow freely, right? There is no additional energy required. So potassium channels. So this is passive uptake. At the same time, chloride ions also, because there has to be some electrical balance, right? So positively charged potassium ions are flowing in, right? Negatively charged chloride ions will also flow in, okay? However, for that you will need energy, right? And that is done through a symporter. Because remember, the hydrogen ion concentration outside is higher That means the natural tendency of the hydrogen ions or protons is to move inward, right? So the chloride ion has to basically piggyback on the hydrogen ion, right, on the proton, and move inside, right? So that's a symporter. Transport in the same direction, right? Secondary transport in the same direction. So, and chloride ions, so chloride ion is taken up by proton chloride symporter. And again, these terms should be clear to you by now because I have already covered them. I have explained in detail, right? So at least for this part of the course, make sure you study all the earlier material before you come, right? So now what happens? So now you have potassium that has entered, you have chloride that has entered. So really you have potassium chloride in solution. What is that going to do to the water potential or osmotic potential inside the cell? Is it going to raise it or is it going to lower it? Huh? You have increased the solute inside the cell, okay? What is that going to do? Lower the osmotic potential, all right? So it lowers the osmotic potential inside the guard cell, right? So what is going to result from that? Osmotic potential is lowered, water potential is lowered. So what is going to happen? Inward flow of water, right? So implies that water is going to flow in. So water flows in and it raises the turgor pressure, right? Or the pressure potential, right? And this is going to cause the stomata to open, right? I mean, there is a mechanism how things are lined up and all that, but in response to more water, this thing is going to open up. So this will cause, causing stomata to open. Right? Now, see, like the potassium concentration in the cell, right, that varies during the day. Early in the morning, right, when the sun rises, right, that time the opening of the stomata is brought about by the potassium, um, uh, the potassium ions, right? But later in the day, it's a different story because like around late afternoon, the potassium goes down. But the stomata still stay open until like about sunset, right? So that means there must be some other solute that is contributing to that, okay? And that basically comes from sucrose, right? The sucrose concentration goes up. As the potassium concentration is going down, sucrose goes up, right? And then around sunset, the sucrose goes down. So that's when, there'll be a loss of turgor pressure and then the stomata close, right? So this happens early in the morning. By early afternoon, the K plus content in the guard cells begins to decrease. But stomata still remain open. This is possible because sucrose concentration goes up. And this can happen because of a number of reasons, right? You could have starch breakdown, right? So sucrose concentration goes up. It could be either because of starch breakdown. Remember I said earlier that starch is insoluble, right? So if you break down starch, you're producing sucrose. That can dissolve in water. It can lower the water potential, right? Keep the stomata open. Another possibility could be, this is the reason, sucrose is also produced because of photosynthesis, right? So during the day photosynthesis is taking place, so in the cytosol, the sucrose concentration could go up, right? So there are many different possibilities, right? So by the end of the day, the sucrose concentration will start going down, right? Because the sun is going to set and all that. So by the end of the day, the sucrose content goes down. That implies that the water potential inside the gut cell, what is going to happen to that? It goes up, yeah. So inside gut cell goes up, which means that water is going to leave the gut cell and causing the stomata to close. So water leaves the gut cell, which means that the gut cell turgor pressure drops and stomata close. Right? Any questions? Is that clear to everybody? Right? There's different steps that are involved. Right? Now, what is the molecule in these gut cells? That basically is the blue light photoreceptor, right? That is sensing the blue light and causing the stomata to open, right? So the blue light photoreceptor in gut cells is a molecule which is called, so in gut cells is mediated by zeaxanthin, right? And what is zeaxanthin? It is like when we were talking about photosynthesis, right? We talked about something called xanthophylls, right? That can take away the excess energy, light energy, right, and dissipate it as heat. So xanthophylls, like they can be in three forms, okay? They can interconvert from one form to the other, depending on the needs of the moment, if energy dissipation has to be carried out. One of them is zeaxanthin, right? So zeaxanthin is a component of the xanthophyll cycle of chloroplasts, which protects photosynthetic pigments from excess excitation energy. So from excess excitation energy. Right, that's why we discussed this when we were discussing photosynthesis, right? But in addition to this, right, in addition to that function, zeaxanthin is also a receptor for blue light, right? So in addition, zeaxanthin functions as a blue light photoreceptor in gut cells, mediating blue light stimulated opening. And indeed, people have done work to see that the action spectrum, that means the range of wavelengths over which this molecule is active, right? So action spectrum of blue light stimulated stomatal opening closely matches the absorption spectrum of this molecule. Also, people have seen that the incident radiation, right? Incident radiation. So if you measure the amount of incident light, right? Incident radiation, then the zeaxanthin content of the guard cells and stomatal apertures, that means the size of the opening, right? They are all closely correlated. So blue light brings about stomatal opening through the action of zeaxanthin, right? Now if you give blue light and then you also give green light, right, then the effect is reversed, right? Because what green light does is it basically converts zeaxanthin into some kind of an isomer, right, and this activity is lost, right? So blue light stimulated opening stimulated, so blue light stimulated opening of stomata can be reversed by green light, and this involves involves inactivation of zeaxanthin via isomerization. So isomerization means same chemical formula, different structural formula, right, and different properties. All right, any questions? So that concludes our discussion of stomatal biology, right? So no questions, then I move on to the next chapter, right? Which, yeah.
So is the green light reflected off of the leaves and it's controlled for the stomatal opening closing?
No, green light is reflected off the leaves because chlorophyll,
No,
right?
it's the chlorophyll, but I'm saying is it like, is that, I guess that seems like it might be some sort of like naturally developed pseudo control of some sort where, incident green light reflected off of a chloroplast inside of something.
Yeah, it is reflected off the chloroplast because the chloroplast is
Green, red,
yeah,
right?
the chlorophyll, right? The chlorophyll basically is going to absorb in the red and blue region, right? And the green region it reflects, right? So yeah, it's everything is kind of knit together, right? And it may be a natural adaptation, Maybe that's how it evolved. I don't know for sure, okay?
It just seems like
Yeah,
an interesting
yeah.
side
It seems like everything works right, okay? So yeah. All right, any other questions? So then I move on to the next chapter, which is chapter 11. And here we are going to talk about translocation in the phloem, right? So recall that plants have two kinds of vascular tissues, right? Xylem and phloem. Xylem is the one that is made up of dead cells, right? Forming all those pipes that bring water and nutrients from the soil, right? And move it up the plant, right? And phloem is basically the one that translocates the products of, because photosynthesis is carried out in the leaves, okay? So the roots and other areas also need nutrition. So phloem is the one that does the translocation, and it is made up of live cells, right? So there's live activity going on. It's not just a bunch of pipes, all right? So phloem translocation moves the products of photosynthesis from mature leaves to areas of growth and storage. And the phloem also transmits chemical signals, right? So also transmits chemical signals. And we'll see more of that later, like plant hormones and so on, right? And redistributes ions and other substances throughout the plant body. Now the phloem is made up of units that are called sieve elements, and let's see what they look like, right? So the phloem is made up of things that are linked together, units that are called sieve elements, and a typical sieve element will look like this, right? It's a tube like this that has got some holes here, larger holes, that will form what is called a sieve plate, right? And then on the side also there will be holes, right, that are called sieve pores, right, lateral sieve pore. Right? And elements like this are linked together, right? Now one difference between this and the xylem is that, this sieve element typically will also have a companion cell. So when the sieve element is formed, there is also a companion cell which does most of the activities, all right? Because the sieve element will lose most of its organelles inside, so it cannot, take care of many of the cellular functions, right? So typically, the sieve element will look like this. It will have like a tube-like structure here with all these pores, right? The sieve plate, and then the same thing at the bottom, right? And then there is a companion cell, right, that it is attached to. There is some other cell here that is called a companion cell, right? And it can communicate with the companion cell through plasmodesmata. Right? And this is a companion cell. And this one is the sieve element. Right? So the two of them are together. The companion cell is going to supply energy and do other things that are needed, right, to the sieve element, because the sieve element has gotten rid of many of its structures. Like this one, this sieve element has gotten rid of nuclei. So no nuclei, right? So no nucleus. Then vacuoles, then Golgi bodies, and ribosomes, right? And this guy, right, so it doesn't have any ribosomes, so it cannot do any protein synthesis, right? So proteins have to come from this companion cell. The companion cell has the machinery for doing protein synthesis, and then ATP generation, so it'll have mitochondria. Okay? And the sieve element, it'll retain some organelles. So like some of the organelles are retained. So sieve element retains some organelles, right? Like modified mitochondria. Then plasma membrane. Then smooth endoplasmic reticulum, etc. Right? The point to note here, you don't need to know what is retained and what is not retained, but it is not capable of doing all the functions, right? It depends on the companion cell to help it out, right? Now, if you're looking at companion cells, they can be of three different types, right? So let me just mention that. So companion cells can be of three different types, right? So types of companion cells. The first type is what are called ordinary companion cells. So these cells have chloroplasts and they have variable number of plasmodesmata, right? Then the second type, these are called transfer cells. And the important feature to note is that they are similar to the companion cells, right? But they have no plasma or very few plasmodesmata, right? And the third type are what are called intermediary cells. They have lots of plasmodesmata, so numerous. Right? So the transfer cells have got very few plasmodesmata. That means if they are communicating with other cells, right, outside, then it has to be mostly what kind of transport? Is it going to be symplastic or apoplastic? Very few plasmodesmata. So that means it has to be apoplastic because you don't have plasmodesmata, right, for carrying out that transport. So this one is mostly apoplastic transport when things are coming from outside. Apoplastic transport. For intermediary cells, they have numerous plasmodesmata, right? So they can communicate with the other cells, right? These companion cells through symplastic transport, right? So symplastic transport. That means where you don't have to cross the plasma membrane, right? Things are linked together. So symplastic transport. And then for the ordinary companion cells, it can be either symplastic or apoplastic, right? So symplastic or apoplastic, right? So just keep this in mind that because later on I'll probably have to get to that, okay? So let me just make sure. Yeah. Later on, just keep in mind that for this guy, right, for intermediary cells, all right, it is only symplastic transport, right? Just keep this in mind. I'll later on need to use that. That's why I showed you the differences between the three, right? So now, and another characteristic that these transfer cells have is that the membrane, right, is very much, indented on the side that is away from the sieve element, right? So basically it tries to increase the surface area on that side for absorption of nutrients, right? Now, if you're looking at these, so you have the sieve elements, right? I mean, these are the sieve elements. If you have them stacked one on top of the other, right, that is going to produce a sieve tube, right? And then the element will be called a sieve tube element, right? Now, in general, so there is, stuff that moves around inside these sieve tube elements, right? That stuff is called the sap, right? Uh, the sieve element sap, right? Like when you puncture that, right? Some fluid comes out. Okay, that's called the sap. But in general, that's hard to study, isolate and study, right? So sieve elements. So sieve element sap in general is hard to study. First of all, it's hard to study non-invasively because if you don't go and puncture that, you're not going to get the sap, right? So you cannot just observe it from outside, right? So sieve element sap hard to study non-invasively, right? And in general, the plant is going to have mechanism because that sap has got sucrose and all other kinds of useful things, okay? So it is going to try to guard that very, heavily. It's going to be guarded very heavily because if it loses too much of sap, that is going to affect the viability of the plant, right? So just like, if you have a cut, your blood clots, right? So there are repair mechanisms in place, right? So, so one of the reasons why it's hard to study this sieve element sap is that damaged sieve elements are sealed off very promptly, right? And and this can happen. There could be either a temporary seal or a permanent seal, right? So this sealing off can happen by what is called referred to as P protein, right? And that is a temporary fix. Right? You can think of it like a blood clot, right? Blood clotting locally at a wound, or so very promptly by P protein and by something that is called callose, right? Which is actually like a polysaccharide, right? It's actually beta 1,3 glucan. That means it's made up of glucose units, right, that are linked together, right, to make this polysaccharide, right, and that's called callose. So it can be sealed off by callose, and this is a longer term solution. Again, the important point to note is that when you try to get this sap, right, you puncture, right, the sieve elements, right, but there's something that in there that is going to try to seal it, okay, because that's the plant's defense against it, right. There are also other problems, right, that are associated with trying to, get the pure sieve element sap, right. So other problems that you will run into. The first one is that when you puncture, right, so you have damaged other cells. So the initial stuff that comes out, that's not going to be pure sieve element sap, okay. It's going to be stuff that is mixed up from the other cells, right. So the initial material with initial material is contaminated with damaged tissue. Then the second thing is that things are under high pressure, right? Remember, in the phloem, things are flowing, there is water, potential and pressure, turgor pressure and so on. So if you puncture, things are going to start coming out, okay? So when that happens, there is loss of turgor pressure. So from the neighboring cells, there might be things that come in, like water and other things, right? So under high pressure, sudden release of this high pressure may bring in contents from other cells. So it will not just be the sap in the sieve element, from other cells. Then the third thing is that this sap or this exudate, that means the thing that is exuded, that the thing that comes out, so the exudate substantially diluted by water flowing in. So substantially diluted by the influx of water from xylem and surrounding cells. Because remember, the xylem is getting water from the ground, okay? There the pressure hasn't changed. Here if there's a loss of pressure, some water will flow in, okay? So xylem and surrounding cells. So one fix for collecting relatively pure sieve element sap, right, is the following, right? So the solution to this is to use what is called an aphid syringe. So aphid stylet, right? And I'll explain in a moment what that is. Use aphid stylet as a natural syringe. So what are aphids? Aphids are insects, right? That are phloem feeders. I mean, have you heard of those aphids? Okay, they can kill your tree, okay? Because they will directly get into the phloem, right? And just take the sucrose and all the nutrients away and they'll survive and they'll kill your plant, right? So aphids are insects that are phloem feeders, right? In fact, earlier this summer, in my house, right, there were three plants like crepe myrtles that got infected with aphids. So they had to come and treat it because I'm seeing the leaves falling off, and all that because they just get into the phloem and start feeding off your plant, right? So these guys are phloem feeders, right? Now the good thing is that they have a syringe, okay? That is not going to mess everything up. That is going to go and pierce only one sieve element and they'll keep on drawing fruit from there, okay? So you can use that, in order to avoid all these other disadvantages, right? So you can take an aphid, put it under anesthesia, cut off its syringe, and then use that to extract fluid from the phloem, right? So the aphid stylet inserts into a single sieve element. So you don't damage neighboring cells, right? And avoid a lot of these other problems that I pointed out. So this method yields relatively pure sap. And then you can study that. All right, any questions? Any questions so far? So what is the pattern of translocation in the phloem, right? It's from the source to the sink. So pattern of translocation in the phloem. This will be from the source to the sink. And this can change with time, right? Like when new leaves are formed, before they have matured, okay, before they have become green, right, and they can do their own photosynthesis, those leaves are actually sinks because they have to take in nutrition from other places in the plant. Once they start doing enough of photosynthesis, okay, more than producing more than what they need, then they can become sources and they can start exporting to other areas of the plant, right? So this can change with time. So this can change with time. Can change with time. Now, what are the materials that are translocated in the phloem, right? So materials translocated in the phloem. The first one is basically sugars, right? Because different parts of the plant, they need carbohydrates for energy. And typically the sugars are going to be non-reducing sugars, right? Non-reducing sugars. That means not sugars which are not very reactive. So non-reducing sugars. And the one that you get, mostly in plant sap is sucrose, right? Now why is that? See, sucrose is made up of what? Anybody remember? And all right, so sucrose is made up of glucose. Okay, so glucose and then fructose, right? That gives you sucrose, right? And what kind of reaction is this? Huh? So it's a no, it's not hydrolysis. Condensation, yeah, because yeah, it's the opposite because you're linking them together. So glucose plus fructose will give you sucrose, right? The problem is that, glucose is very reactive because glucose is what? Is it an aldehyde or a ketone? Aldehyde, yeah. Right? And fructose is a ketone, right? And you can check that if you look back in the earlier chapters, you will see that. These guys are reactive, right? So if they're transmitted as sugars in the phloem, right, they will react with different parts of the plant, right? Undergo reactions. Whereas with sucrose, you don't have that problem, right? So, but animals will see in our blood, right? What kind of sugar do we have? It's glucose, right? But the concentration is very low, right? That's why it's okay, right? So animals are able to use glucose, right, as their energy source and transmit it from one place to the other because it's at a very low concentration. Inside the plants, the sugar will be at a much higher concentration. So the potential to react with other things in the plant is a lot higher. So in the case of plants, it's sucrose, right? So glucose plus fructose is sucrose. Then you can add a galactose to sucrose, right? So sucrose plus galactose. And of course, I'm not giving you the details about the location at which it has to be added because, these are again, if you're looking at glucose, you have 6 carbons in there. So there are different places where that condensation reaction can take place. But sucrose plus galactose gives you another sugar that is called raffinose, right? Similarly, raffinose plus galactose gives you another non-reducing sugar that is called stachyose, right? And stachyose plus galactose gives you yet another non-reducing sugar which is called verbascose, right? And if you look at the sap, right, if you look at the sieve element sap in plants in the phloem, all these sugars will be there. Sucrose is there, raffinose is there, stachyose is there, verbascose is there, right? These guys don't react. Okay, so these are things that you will find in the phloem sap, right? Right, yeah. So these are all commonly translocated in the phloem, right? All commonly translocated in the phloem. So in addition to that, you will have, right, so these are the kinds of carbohydrates that you will find in the phloem, right? In addition to that, you will have amino acids, and typically you will have important amino acids are glutamate and aspartate. Remember, we talked about 20 different amino acids. We didn't study their structure, but there are 20 of them. 2 of them are glutamate and aspartate, and their amides, respective amides, which are called glutamine and asparagine. Respective amides, which are called glutamine and asparagine. Right? These are also amino acids, right? But they are related to each other. So if you have glutamate, right, you add an amino group to it, right, you get glutamine, right? If you have aspartate, you add another amino group to it. Aspartate is already an amino acid, but you add another amino group again at a particular location. Let's not worry about that. But then you get asparagine, right? This is important, like going from glutamate to glutamine, all right, aspartate to asparagine. That's important because even in our bodies as well as in plants, that's how nitrogen is transported, right? There are reactions that are called deamination, transamination reactions, and so on, which I will talk about a little bit later, right? So respective amides, glutamine and asparagine. So typically in absorption of nitrogen, these are the guys that are initially produced. Then the other amino acids you get by transferring an amino group either from a glutamate or an aspartate to something else. Okay. Then in addition you will have RNAs, then you'll have proteins. For example, the P protein that is working like the clotting factor, right? Example, P proteins, right? And then you will also have what are called plant hormones, and I will talk about these later on, such as auxin, then gibberellins, cytokinin, and what is called abscisic acid. Don't worry about memorizing these, okay, auxin, gibberellin and all that stuff, because I'm going to discuss these later. Okay, I don't know in how much detail, but they'll be mentioned again later on. Now it is a fact that the velocity of movement in the phloem is about 1 meter per hour, right? And this is a lot faster than diffusion, okay? So lot orders of magnitude faster than diffusion. So what is the mechanism by which transportation takes place in the phloem, right? One of the proposed models is what is called the pressure flow model. Of phloem transport. So recall that we have this equation for the water potential, right? Psi equals psi s plus psi p, right? Where psi s was the solute potential, right? Psi p was the pressure potential, right? So at the source, what is going to happen? See, at the source, let's say the leaves are producing sugars, okay, let's say sucrose, right? That's getting dissolved in uh, water, right? It is going to lower the water potential, right? That means water is going to rush in from outside. That is going to cause the turgor pressure to go up, right? So at the source, accumulation of sugars in sieve elements so this so in sieve elements generates negative solute potential, implies water potential falls. That means water is going to enter. So water enters the sieve elements, which means the turgor pressure is going to go up. So that's what happens at the source, right? Now at the sink, just the reverse will take place. So at the sink, the unloading, right, at the sink of the stuff that is being carried, right? So phloem unloading causes turgor pressure to drop. So the flow, so the turgor pressure at the sink is lower, at the source it's higher. So the pressure gradient is what is going to drive the flow, right? So flow from source to sink driven by pressure gradient. Now there are some observations, right, that support this pressure flow model, right? So observations. Nobody knows for sure whether this is the correct model or not. Okay, there are other models that have also been proposed. So observations supporting the pressure flow model. One, there is unidirectional transport in the phloem, all right? You don't have things flowing in two directions, right? So in the same sieve element, you do not have molecules flowing in two directions. They are only going in one direction. So unidirectional flow, because if it's the pressure gradient, that can be only in one direction, right? Then the second thing is that both the solutes and water move at the same speed. Move at the same velocity. And I just note that other models have also been proposed, right? So other models for translocation have also been proposed.
Are they not certain that that's how it works?
No, they are not certain, because this model cannot explain everything, all right? So also been proposed, yeah. But again, I mean, I don't have time to discuss all of that stuff, because we are not plant biologists. Yeah, no, they are not. Okay. All right. Now let's look at cargo loading at the source and unloading at the sinks. Right? So cargo loading at source. So what are the steps involved? Step number 1, right? The sucrose produced in what are called the mesophyll cells. Not, the sucrose is not produced in the companion cell or in the sieve element. Okay, it's produced somewhere else. It has to get transported over there. So sucrose produced in mesophyll cell in the leaves. Step number 2, sucrose moves from cells in the mesophyll to cells in the vicinity of the sieve elements in the smallest veins of the leaf. And this is called short distance transport because it's transportation only over short distances, right? Long distance transport is transportation through the phloem, right? It will go maybe from the leaves all the way up to the roots and so on. So short distance transport and step number 3. So this has come close to the sieve element companion cell complex, all right? Now it's going to get loaded, so it's called phloem loading. So phloem loading. So this is sucrose loaded into sieve and companion cells. Right? And this can be achieved, right? Remember, I talked about those different types of companion cells, some of them symplastic transport, some of them apoplastic, right? So it can be loaded in different ways, all right? So it could be loaded via the apoplast, right? So there could be apoplastic loading. And that happens through the use of a proton sucrose symporter, right? Remember, outside the cell, right, in the apoplastic region, the pH is higher, right? So hydrogen ions have a tendency, or protons have a tendency to move in, right? So the sucrose can just hitch a ride with them, right? So hydrogen ion, so proton sucrose symporter is going to do that. And the other one is the transportation through the symplast, right, or symplastic loading. And this is via plasmodesmata. And for this, right, remember we said that among those 3 different types of companion cells. The intermediate companion cells, they have a lot of plasmodesmata, right? So what is going to happen is that, so those are the ones that will be having more of symplastic transport. So the sucrose enters intermediary type companion cells. So it is, and then once it gets in there, okay, it gets converted into this raffinose and those other things that I talked about, right? So converted into raffinose, etc. Right? Now raffinose is a larger molecule than sucrose, all right? So you have in these companion cells, in these intermediate companion cells, all right, you have plasmodesmata that are, that are basically going into the sieve elements, all right, and others that are going into those other cells, from where the stuff has come in. But if you now have a larger molecule, it cannot diffuse out through that smaller size pore, right, plasmodesmata. It can go only in one direction to the sieve elements, right? So converted into raffinose, etc. So these are larger molecules. So they can only go to sieve element. And also by converting sucrose to something else, you're maintaining the concentration gradient, right? So can only go to sieve element. Also sucrose concentration gradient is maintained. Right? And this is referred to as the polymer trapping model of apoplastic transport. Because you're starting with sucrose, it's going in, right? Then it becomes a larger polymer, then it's trapped over there, it can go only in one direction. So it's like a street, suddenly it becomes one way, so you cannot go back, right? So polymer trapping model of symplastic transport. Right, so this is how the how the stuff is loaded into the phloem, right? Then it will go, and then there has to be unloading. It's just these steps in reverse, all right? So the importing at the sinks involves the following steps. So import at sinks. Step number 1 is phloem unloading. Then there'll be some short distance transport because the sink might be some way off from the phloem, right? So some short distance transport. And then at the sink, there will be storage and metabolism in sink cells. And this phloem unloading and then this short distance transport, it can be symplastic or apoplastic. And here apoplastic transport is going to be down the concentration gradient because in the phloem it's higher, the concentration. So you will not really need a symporter, right? It will be down the concentration gradient, so it can be diffusion or something else, right? Now this kind of transportation will be apoplastic, right? In the case of seeds, right? Why? Why? Because there are no symplastic connections between the maternal tissue and the seed tissue, right? Because no symplastic connections between maternal tissues and tissues of the embryo. Now, as I said before, the, the transportation is from source to sink, all right? But the sink can get converted into a source as the leaves mature and so on. Okay, so just keep that in mind, right? All right, any questions? Any questions? All right, so then I will move on to the next chapter, right? I'm trying to give you the big picture, right? I could have skipped some of these topics, but I thought that, since I spend the time, understanding that stuff and from an engineering point of view, making sense, so I'll just give it to you, even if it takes a little bit of time. So we want to move on to the next chapter, right, which is about respiration and lipid metabolism, right? So chapter 12, which is respiration and lipid metabolism. And you'll see a lot of things that will come together, right? Because we have already discussed photosynthesis, now we'll talk about respiration, right? So in respiration, if you're looking at animals, all right, the overall reaction is, so in animals, the molecule that is used is glucose, all right? So C6H12O6, right, plus 6 oxygen, 6 molecules of oxygen will give you 6 molecules of water plus 6 molecules of carbon dioxide, right? And this is in animals. And a lot of energy is released, and it has to be a controlled reaction, step by step, right? Because if all that energy is released, it's going to be released as heat, right? You don't want that. You want to capture that energy in ATP and in other high energy molecules like NADH and so on, okay? So this is in the case of animals, so this is glucose, all right? In the case of plants, it is going to be sucrose, right? Not glucose, all right, sucrose. So the overall reaction for respiration is C12H22O11 plus 12 oxygen will give you 12 molecules of carbon dioxide plus 11 molecules of water, right? Because remember, sucrose is produced from glucose and fructose in a condensation reaction. So one molecule of water is already gone over there, right? So this is sucrose, right? So this is in plants, right? Now the good thing is that when you're studying respiration, right, I said that it involves many different steps, okay? So naturally, you know that sucrose is made up of glucose and fructose, right? So the first step would be to break up sucrose into glucose and fructose, right? And then at some point in time, right, actually it's after 1 or 2 steps, the pathway will be exactly the same in plant and in animals. I shouldn't say exactly, there might be some subtle differences here and there, but more or less it's going to be the same, right? So what I covered here is going to also apply to respiration in animals also, okay? So one important point which I pointed out a long time back is that these reactions have to be carried out at a very controlled rate, right? So that small amounts of energy are released and they can be absorbed. True in plants, true in animals also, right? So the first step, right, if it is in the case of plants, right, from sucrose you could break that up. This is not by any means the only way in which it can happen, but you could split up sucrose into glucose, right, and you could split it up into glucose and fructose, all right. Now you can put a phosphate on it, right? You can phosphate, if you phosphorylate glucose, right, you're going to get glucose 6-phosphate, right? That means a phosphate on carbon number 6, all right? Or you could do fructose. If you put a phosphate on carbon number 6, you can get fructose 6-phosphate, right? And these are actually isomers of each other. So there are enzymes that will help you to convert from glucose to fructose and vice versa, right? So you can go from one to the other, right? Now, if you look at respiration, whether it's in animals or in plants, okay, there are a number of steps that are involved, okay, in the oxidation of, let's say, glucose or fructose, right? So the steps are as follows, all right, and you can study them separately, all right? Everything basically meshes together. The first one that we will look at is called glycolysis, right? Glycolysis means basically splitting of sugar, right? See, if you look at glucose, it's a 6-carbon sugar. If you do glycolysis, you will split it up into 2 3-carbon sugars, right? So, and this occurs in the cytosol, right? Whether it's in plants or in animals, this occurs in the cytosol. Another one procedure, right, is called the pentose phosphate pathway, And this is the pathway that, so remember, glucose is a 6 carbon sugar, right? Now if you look at DNA and RNA, what sugar do you have? Huh? Ribose or deoxyribose, those are 5 carbon sugars. Those are pentoses, all right? So you need some pathway to go from glucose to pentose, right? And that is the pentose phosphate pathway, which I will discuss very briefly, right? So glycolysis, pentose phosphate pathway, then you have something that is called the TCA cycle or the Krebs cycle. This occurs in the mitochondria of cells, right? Mitochondria of cells, that's the location. And then the last one, which results from the TCA cycle, is called the oxidative phosphorylation or production of ATP. This will be very similar to what we discussed in photosynthesis, all right? So oxidative phosphorylation, the same idea. You build up a proton gradient, then when protons move down that gradient, there's something that is churning and producing ATP, right? So again, I mean, it was worthwhile spending time before because it will take me less time right now, right? And I can connect up everything together. So oxidative, we won't finish it today because there are a lot of things involved, all right? But I'll try to at least make some progress, all right? So oxidative phosphorylation, right? So the glycolysis is the splitting of sugar, right? Let's say glucose, all right? You could start from sucrose also, but you'll split it up into 2, 3 carbon sugars, all right? Glycolysis does not use any oxygen, all right? No oxygen used, right? Oxygen used. The TCA cycle is where oxygen is used, all right? Or actually oxidative phosphorylation is where the oxygen is used, all right? So this is the one where oxygen is used, all right? Glycolysis produces a very small amount of energy. Let's say from 1 molecule of glucose, all right, you'll probably get 2 ATPs, all right? If you run it all the way through from glycolysis to the TCA cycle, right, and then through oxidative phosphorylation, you get 30 molecules of ATP, right? So using oxygen gets you lots more energy, right? So first let's look at glycolysis, right? Right. So let's say you had fructose, right? In the case of plants, right? You have converted it into from fructose, right? You went to fructose 6-phosphate, let's say, right? Now, obviously, the phosphate has to come from somewhere. So where would that come from? ATP, right? And again, I mean, something that adds a phosphate, that is going to be what kind of an enzyme? Some kind of kinase, all right? So some kinase. Again, you can look at the book. All those details are given, right? But I'm trying to give you the big picture and, without having to memorize all the details. So some kinase is there, right? All right? That'll take you from fructose to fructose 6-phosphate, right? And this is the point. Fructose 6-phosphate is the point where glycolysis starts both in plants as well as in animals. In the case of animals, glucose is going to be phosphorylated to glucose 6-phosphate. Then some isomerase is going to convert it into fructose 6-phosphate. In the case of plants, I mean, there are a couple of different pathways that will get you to that same point, fructose 6-phosphate, right? And once it gets there, then your glycolysis is starting, or a full-blown, whether it's in plants or in animals. The first step of that is there is an enzyme that is called phosphofructokinase, right? Right? So what is that enzyme going to do? I have fructose 6-phosphate, all right? From the name of this enzyme, all right, it's called phosphofructokinase, all right? It's a kinase, so it's going to add one phosphate group, all right? So it is going to need ATP to get that phosphate group from, okay? Because remember, all these reactions are energetically unfavorable, all right? Or many of them are, okay? Well, I shouldn't say all because some of them are going to be favorable because you're now doing respiration, okay? It's not photosynthesis, all right? But this reaction is energetically unfavorable, so it requires ATP, all right? Requires ATP. And so now you're going to get, you already had fructose 6-phosphate at location number 6. Now you're going to have 2 locations where you're going to have phosphates, all right? So you, instead of calling it a phosphate, you're going to call it a bisphosphate, 2 locations, all right? And which locations are you going to have the phosphate? Those will enter into the name, right? So instead of fructose 6-phosphate, it's going to be fructose 1,6 bisphosphate, right? So fructose 1,6 bisphosphate, right? Then So you now have two phosphates, okay, on this fructose molecule. You can break it up, right, into what are called triose phosphates, because this is a hexose phosphate, all right? It has got 6 carbons, all right? You'll break it up into triose phosphates, all right? And there are 2 of them. And again, these are isomers of each other. So you will get what is called glyceraldehyde, 3-phosphate, right? And then the other one is called dihydroxyacetone phosphate. And if you have the time, right, or the energy, you can go and look in the book to make sure the first one has an aldehyde. So it should have an aldehyde group, all right? The dihydroxyacetone, that should be a ketone, right? So there'll be a ketone group in there, right? If you look at the detailed formula. I'm just saying that everything makes sense, right? But you don't have to memorize anything, right? So these are triose phosphates, right? Have you seen these things before? Have you seen like glyceraldehyde 3-phosphate before? Where? In? In one of the reactions in what? Photosynthesis, right? We've seen that before, right? When we did those carbon fixation reactions, right? So now in the next step, right, this glyceraldehyde 3-phosphate, right, will get converted. So this is actually, some of these reactions are reversible, all right? It will get converted to what is called 1,3-bis phosphoglycerate, right? And this reaction requires an inorganic phosphate to go in, right? And it also requires NAD plus, right, to go to NADH, right? Some electrons are given out and the high energy electrons are stored in NADH, right? So you go to 1,3-bis phosphoglycerate. Next, the next reaction is what is called a phosphoglycerate kinase reaction, right? And that gives you 3-phosphoglycerate. Have you seen this before? 3-phosphoglycerate, have you seen that? Anybody? Where? Not you, somebody else. Where do you see it? This was one of the molecules that you saw in the Calvin-Benson cycle, right? When we did photosynthesis, right? So that means the product from the Calvin-Benson cycle, right? If it were to undergo glycolysis, it doesn't have to come all the way from glucose or fructose, all right? That stuff could be imported in here, right? If it's at the right location. See, all these pathways are actually interconnected, right? So this gives you 3-phosphoglycerate, and the enzyme that makes this happen is phosphoglycerate kinase, right? Then you have a number of steps, or a few other steps, but the final product that you get, okay, and that one is important, right? Other steps in between, I'm leaving them out, because it's like something is converted to something else. You don't have time to memorize all that. It's good to know, but you don't have to. But the next product that you get is what is called phosphoenol pyruvate. Phosphoenol pyruvate. Sorry. And it is abbreviated as PEP, PEP, phosphoenol pyruvate, right? And then this undergoes what is called a pyruvate kinase reaction, right? So pyruvate kinase reaction, right? And what happens here is that you have ADP, right? It is phosphorylated to ATP. ATP is generated. This is called substrate level phosphorylation. No oxygen is involved, right? ADP goes to ATP, right? So you started out with phosphoenol pyruvate, right? So that has got a pyruvate with some phosphate group on it, right? That is going to go to pyruvate. This is going to lose it because that's a very high energy bond, right? That is used to drive the synthesis of ATP, right? And there is one other step in this also where ATP was produced. I'm not discussing that. But this step I'm putting in because this one is important. I need to use it for the subsequent discussion. So from here you ended up with what is called pyruvate, right? So everything is being done without any oxygen, right? Now look, where are we going to run into problems? See, you are using, see along the way I have converted NAD plus into NADH, right? So if you want to keep the glycolysis running, you have to make sure that NAD plus is regenerated. Okay. So how is NAD plus going to be regenerated? You have a number of options from this point onwards, right? Number of possibilities are there. One possibility is if there is no oxygen, right, then you will have fermentation reactions, all right? That will be either produce, and these happen in the cytosol, all right? That will either produce alcohol, right, or it will produce lactate, right? So let me first discuss that, right? And I'll just discuss it right now in the next few minutes. I know we are a little bit over time, but since this figure is there, it'll be easier for me to discuss it, and you can also think about it. So, so there are a number of possibilities. So the first possibility is if there is no oxygen, right, you can see pyruvate is a 3-carbon molecule, right? So 3 carbons in it, right? So the first possibility is that you can remove carbon from pyruvate, right? Carbon dioxide is removed, so minus carbon dioxide, and the enzyme that does that is called pyruvate decarboxylase. And once you do that, you get acetaldehyde. Acetaldehyde has got only 2 carbons, right? There's CH3 and then something with the aldehyde group, C double bonded with OH. And then in the next step, right, you produce alcohol, right? So this is a reversible reaction, right? And you produce ethanol. And this reaction basically takes up NADH and gives you NAD plus. Because you need to regenerate that NAD plus to keep the reaction going. Right? Otherwise the supply of NAD plus will get used up and glycolysis will stop. Right? So if there is no oxygen present, right, then this pyruvate is going to be first decarboxylated to an acetaldehyde, right? And then it is going to be going to ethanol, right? And the enzyme that does that is called alcohol dehydrogenase. Right? Now, if somebody consumes alcohol, right, that is ethanol, that's ethyl alcohol, right? So how is that metabolized? Well, that ethanol first, that alcohol dehydrogenase, because this is a reversible reaction, that is going to give you acetaldehyde, right? In the liver, extremely toxic, okay? Aldehyde is extremely toxic, right? That's why, excessive drinking causes liver damage, right? So that's one possibility. The other possibility is in the absence of oxygen, right? There is an enzyme that is called lactate dehydrogenase that will give you lactic acid. So it gives you lactate. So this is lactate dehydrogenase. And this gives you lactate, right? So that can cause the environment to become more acidic, right? So these reactions, the ones which produce lactate and the one which produces ethanol, these are called fermentation reactions without any oxygen, right? What is going to happen with oxygen, I will talk about next time, okay? Because then the pyruvate has to move into the mitochondria and, with the use of oxygen, a lot more energy will be generated. This glycolysis pathway gives you only 2 molecules of ATP per molecule of glucose. Or if you start with a molecule of sucrose, you'll get 4 molecules of ATP. Very, very energetically inefficient. But in the worst case, at least the cells can survive. Because if you have flooding, okay, in the roots, no oxygen is getting there, right? So it can use the fermentation reactions, produce alcohol and survive, right? And this is how, all this wine and all that is produced through fermentation, right? In an oxygen limited environment, yeah. Let's stop here for today, yeah.
I was going to say that also is what happens when you exercise when your muscles get sore is from lactic acid.
Yes.